All right, our next speaker is kindly standing in for Thomas Fuchs. Again, thanks very much to Michael Becich. So Michael is Distinguished Professor of Pathology and Chair of Biomedical Informatics and Associate Vice Chancellor for Health Informatics at University of Pittsburgh School of Medicine, and leads many of the health IT innovation efforts there. He will be speaking to us this morning on using AI to enhance pathology diagnosis. So Michael, thank you.
All right, thank you very much. Appreciate the dual opportunity this workshop's provided. So I'm a basic science chair in a medical school. And unlike a lot of my informatics colleagues that are very clinically facing, we're trying to push the ball up the hill in diagnostics through research and discovery. So a lot of what I'm going to talk about today are really NIH and particularly NCI-supported efforts that are trying to democratize imaging data to the common purpose that both Amy and Kurt have talked about. So I do have a good number of disclosures. I tell my faculty that they should have slides that even have more than this on it, and we're making steady progress on that. I will talk about two technologies that my department and I personally benefit from, potentially. The first is a DID algorithm our team developed a while back with an IAMS grant from the NLM that is in commercial use by a lot of our partners. And the second is a startup in this space, pathology AI company called Spintelix. And I will mention them in passing. So those are significant financial conflicts of interest. I do a lot of consulting at cancer centers and academic health centers, and I have a good array of federal funding, which is really the bootstrapping of all of the efforts I'm going to talk about today. So the take-home messages I put up front here. Why is pathology a rich space for thinking about AI in partnership with radiology is that we're catching up. FDA has now approved whole slide imaging, essentially an automated platform for robotic capture of histopathologic cancer diagnostic images. Philips got FDA approval, and there are 3 companies in the process of getting FDA approval. And so much like radiology did a decade and a half ago, there is instrumentation that the FDA has approved that will be put in laboratories to use for primary diagnostics. Today, a lot of secondary consultation and research and teaching support happens on whole slide platforms, but now we're going to have them essentially in the primary labs serving our cancer patients. As a biomedical informatician, I've got to put the informatics word in there, so I think informatics is key to implementing what we're calling in pathology as computational pathology and bringing AI into clinical practice. And I'll talk about how from the R&D end we're going to approach that. And I'm going to stress, which I think is a key opportunity, certainly radiology has been here a lot longer than pathology is, how pathology and radiology can partner. And can we put together some information technology, informatics platforms to enable AI? So I'm going to talk a little bit about some of the research efforts in Pittsburgh, particularly a natural language processing platform for pathology and radiology reports, and an image sharing platform called the TIES Cancer Research Network, which the NCI supports, which will then allow very highly annotated images to be shared as open data among cancer centers. And then finally, drive deeply to how do we create shared federated repositories that are fair for the cancer research community. The NCI has an effort called the Cancer Image Archive, and we're partnering with the TCIA to, like with TCGA, democratize imaging data for cancer. So it's an interesting time. So whole slide imaging workflow is shown here. This part doesn't change. This is the pathology lab, hand by hand, taking pieces of tissue, putting them in cassettes, and turning into paraffin blocks. And the classic workflow takes it through quality checking and sectioning, and pathologists then distribute caseload and look at it with analog microscopes, which have no standards. In whole slide imaging workflow, cases are entered into robotic scanners The images then sorted into cases, they're reviewed for quality assurance and then distributed to pathologists digitally, just like workflows and PACS systems for radiology. So this is the change that's coming in pathology laboratories. So kind of the watershed event is this, and the whole march to whole slide imaging, my first two companies in this space were tilted at getting the first commercial whole slide imaging devices, and 17 years later now, we have FDA approval for whole slide images for primary diagnostics, and this guy right here, Mike Feldman from Penn's a hero. He did the large classification studies that actually led to the FDA approval, and this is the abstract at the USCAP meeting where Mike presented the work that ended up helping Philips validate whole slide imaging for their platform. And this is from our lead in pathology informatics at Pittsburgh, Laurent Pantano. It's his talk on FDA and whole slide imaging, the regulatory march that we've been on. So now that we're here, what does that mean? Well, it's going to take a while for most practices to start to integrate this into primary diagnostics, but while that's happening, it really is an opportunity to think about the data, since we're in the era of big, Can I go backwards? I guess I can't. Can you take me back one? It's not. There we go. To give you an idea, Kurt talked about ImageNet. If you took all of the ImageNet data, that would be equivalent to 474 whole slide images. Very robust data set. So, and if you took one largest size, we saw the whole slide images of a pathology transected in a previous talk. If you took the CIFAR data set, another very robust data set, it would actually cover in a spatial map that much. So we're really talking about big data here. So this big data in pathology, which is there, is very rich. There's a lot of features and a lot of information that can be extracted from this. And just like in radiology, there's a cognitive process that we have to train people on that is inherently unsafe, as we saw in some of the presentations early. So the goal in this should be to take and do the R&D necessary to do the discovery of using the data that we're going to have, but also take and adapt it to clinical practice. And that's the march we're on. So we defined computational pathology. A group of us got together. Dave Lewis at MGH led a group of us to kind of say, what do we really mean by computational pathology? It's an approach to diagnosis that incorporates all the sources of data in anatomic pathology and clinical pathology. It takes that clinical data and acts on it. So you take data to knowledge, which is part of the engineering process that, pathologists don't know it, but they're turning into an engineering discipline, to then provide the advanced decision support we've heard about, finally then to take and create from an observational discipline, a knowledge engineering discipline. And the databases that you hear about from Jeremy Warner in the next talk are another critical, important part of the equation. So what we're on a march to do is take the basic science of biomedical informatics and apply it to this clinical discipline of pathology and radiology. And so a lot of the tools we use in informatics, natural language processing, the identification of ontologies, building data models, and automatically at scale de-identifying reports are the research efforts that I'm going to mention today that will help enable this democratized data sharing between pathology and radiology. So we have a system in Pittsburgh called TIES, and I'm not going to be able to take you through the dizzying, mind-numbing informatics pieces that this architecture provides, but TIES is an NLP an information retrieval system that takes and automatically in a pipeline de-identifies pathology and radiology reports and makes them searchable. Because, after all, today, we're all addicted to search. If you can't search in a healthcare context, you're lost. And we know how well electronic medical records allow us to search. So the idea is to create a platform that starts to nibble at these very tough issues of going right into the clinical source documents, bringing them out and making them available for research. It's a system for indexing research resources, so paraffin blocks, fresh frozen tissue, whole slide images can be indexed, and data and annotations can then be shared on a data platform that's both federated and closed for research systems, and open if you then take the data and wrap it as fair data, and I'll talk about fair data in a minute. So we have this as a platform that the NCI supported us to support data, biospecimen, and both pathology and radiology images in the current TIE system. We have 27 million radiology reports from 6 cancer centers, and we have over 5.3 million pathology reports that are being shared and exchanged data, and researchers at those 6 institutions can look at each other's pathology reports, search them, find cohorts, share and exchange this data today. And as part of an effort to kind of expand the network, I have 13 cancer centers, a couple of cancer center directors in the audience here, have provided letters of support to actually expand to the network. So this is scaling. I will not definitely for this group go through the mind-numbing architecture, but I got to show you it's a complex system. There's a firewalled part for identified data. You can do identified searches at your institution, so there's a big advantage. You can actually search your own data in the same way in an identified sense. If you're behind your own firewall, you then share it into a demilitarized zone where de-identified ports, and you can federate across systems. So it's a robust system. Rebecca Jacobson, who's a faculty member in our department, developed the system over the last 10 years. And it's a real gift to the cancer research community because we've been nibbling at this for quite some time, and now we're ready to scale. So what TIES enables is cancer research networks to form. So our research network for data sharing is called the TIES Cancer Research Network. The partners in this are Pitt and UPMC, Stony Brook, Abramson Cancer Center at Penn, Roswell Park, Georgia Cancer Center, and Thomas Jefferson University. And we have 2 sites that we'll be expanding to shortly. And we have another 13 sites that have given us letters of support to try to expand the network. So we are ready to take this to scale. And essentially, we actually can take other forms of computational pathology algorithms and add annotations to images through our portal. And the goal is actually to share this out to open data collections like the Cancer Image Atlas Network. So stay posted. We're going to scale with our network, and we're interested in partners. We're looking at diverse populations of patients to add to this, and I think this will be a very valuable resource for both pathologists and radiologists to work together. Now, the talk today is supposed to be about artificial intelligence too, and its impact, and all of the things that Kurt nicely outlined about how you have to have annotated images, and you have to have a searchable database, and you've got to get clinical data joined with images, and you have to make it all searchable, is a really important part of the work that we're doing. But unless you can apply intelligence engines to it, like we've done with Bayesian algorithms in our Causal Modeling and Discovery Center, this is a big data to knowledge center led by Greg Cooper in our group, you don't have the intelligence engine behind it to sort this out. So our Center for Causal Modeling and Discovery, Kurt's nice stack of showing what AI was and machine learning and deep and neural networks, there's a component of it where you put tools together in discovery environments and use both biostatistical and machine learning methods to actually tease out and experiment with the data to create information models like systems maps for how genes interact, or imaging annotations that make sense for prostate cancer. And these models evolve in these causal discovery algorithms. I won't have time to talk about the projects today, but 2 projects, one focused on signaling pathways using TCGA data, has been enabled by this BD2K funding that has actually led to an algorithm that we believe, and are now validating, identifies tumor driver mutations in cancers, and also can identify the interactions between immune and oncology pathways for identifying therapies for cancer cells, for cancer patients. And then we've used computational pathology and imaging in idiopathic pulmonary fibrosis to do the same thing now, except with imaging and genomic data. So taking the data as sorted, putting these rich annotations on it, and creating this environment has really been the goal of our work in Pittsburgh. So causal discovery algorithms have impact. Here's a paper actually from 2010, an early implementation that actually allowed the driving mutations in melanoma to be identified with these causal discovery tools. So this is a powerful set of tools that we will continue to leverage along paths that have meaning to cancer patients, but this is really discovery science and not clinical implementation. So, Bill, this is a slide that I talked to you about. This is from Bill Stead from 2009. This is the problem we're facing. This is a very nice illustration. Clinicians can handle 5, maybe up to, the really good clinicians, up to 10 bits of data and keep track of them longitudinally and make decisions. But once they get beyond that scale, and certainly we are already in this framework here, we have just overwhelmed, swamped cognitive ability to actually measure this. And this is why these algorithms applied to these large data sets are important for us as clinicians and as discovery scientists. So I have trouble telling you where we're headed, so I'm going to just give you sneak peeks. We really believe this platform can help us solve some of the big problems in cancer. So we use spatial heterogeneity in our machine learning algorithms. This is work that was reviewed in Nature Reviews of Cancer in 2009. But cancers aren't just a tumor. They're a tumor in a microenvironment. And today, immuno-oncology is a real driving piece of the landscape. We want to understand how the interactions between the immune system and tumor cells with the vasculature and the framework of the tumor microenvironment are important. Machine learning is going to help us do this. Pathologists don't do this in diagnostics today. Having this digital platform there is an opportunity to give pathologists deeper tools at imaging at scale to help make this. So our approach is actually going to have some scientific impact. Obviously, one of the biggest challenges in cancer is understanding what role heterogeneity plays in diagnostics, what it plays in prognostics and immunotherapy. The NCI has really got a quite exciting project, the Human Tumor Atlas Network, that is going to try to tackle this in the same way that TCGA provided tools for genome. And we think this platform is going to be very useful in that march. And discovery science will be an important part of the contributions of this platform as well. So, pathologists today generally work with H&E and transmitted light. We think it's really important to start to move into multiplexed imaging. There are a number of vendors that have provided tools now that allow you on paraffin-embedded tissues, that's the kind of currency of the anatomic pathology laboratory, to put multiplexed images in place and actually map out all of these cell components. GE has a platform, Perkin-Elmer has a platform. There are new platforms that actually generated work that came out of Vanderbilt initially using proteomic-based images, the MBBI platform, that will actually allow proteomic discovery to occur, protein-level discovery to occur in the same method. But these multiplexed imaging are real good and important discovery tools that we think are going to make it into the clinical pathway, because we'll do a lot with genomics, and then we'll want to reduce it to practice, and we'll want to do it at low scale. They talked about the IBRIS platform and the Oncotype DX in earlier discussions about how a $4,000 test has been reduced to less than $100 by using quantitative imaging. We do think that this kind of platform technology is going to help reduce to practice and quickly for important decision-making in oncology. So mixing multi and hyperplexed imaging with H&E and having them co-registered on a platform is an important part of what we'll be focused on. And we think there are other omics coming too that can be combined on this histopathologic platform. We're starting to see epigenomics come in play. TCGA did genomics, variant calling, and expression data, but epigenomics has not been that tightly integrated yet. We think proteomics is coming. The MBBI platform is an example, and then there's metabolomics. So we want to craft a meaningful pathway, again, with the image of the cancer patient sample at the center of it for research and discovery, and this is part of the path that we're on at Pittsburgh. Ground truth annotation is critical. This is where machine learning can shine and do annotations millions of annotations on individual images in an automated fashion to very high fidelity. This will be important because you need these ground truth annotations to train the models, and then the trained models to increase your specificity of calls. Our platform will help to enable this by using spatial heterogeneity and AI together. One of the important things from a patient safety perspective is to be able to call out actionable regions of interest to pathologists so they don't miss them. Anyone who's reviewed GI biopsies or a prostate biopsy know that these corkscrew-shaped images placed into blocks, 5 to 6 of them just to save cost, means that somebody might miss the important core with the cancer. We think region of interest identification in an automated sense to say, pathologist, hey, here's an area you got to look at. And before your eyes leave a screen or you view it on an imager, to say, you missed this piece, go back to it. We're not letting you move on until you see this. There's a region of interest. Not to do it automated, not to put the pathologist out of business, but to make sure regions of interest are scanned. And then finally, triaging regional interest. Atypical breast lesions are problematic. In situ lesions are problematic. And essentially creating a scale of regions of interest and triaging them to feed up to the pathologist. And then, obviously, these tools will all be fueled by AI. So it's a very exciting time in pathology, but it's even a more exciting time if we get the radiologists to work together. This is one of the slides I found on the internet from Tom's talk. We think computational pathology, genomics, and radiology have an awesome partnership going. A lot of research opportunity, a lot of clinical opportunity, a lot of opportunity to make things better for cancer patients. So with that, I want to thank you for organizing the NCPF. I want to plug the Association for Pathology Informatics, which we've been at for almost 20 years now. We have our own online journal. And I want to particularly thank the team at Pittsburgh that's behind the AI and spatial heterogeneity studies, Chakra Chinabatla, and particularly Chakra and Barak. We have all of our computational pathology lectures online available to folks who want to follow what we're doing in Pittsburgh. We're actively looking for other speakers to come in and talk to us about our work, and we have now 10% of our talks in the radiology space, Kurt. So with that, I will pause.